the rate of electron transport. And electron transport to oxygen is what we think of as respiration. So the rate of respiration is kind of controlled by ADP availability. So this ADP-ATP ratio limits the rate of oxygen consumption in electron transport. This couples the ATP synthesis process directly to the energy needs of the cell. So if you have, you'd never have this, but let's say you imagine some extreme case where the cell's not doing a thing, and it just phosphorylates all the existing ADP to ATP. There's no ADP available. That cell, because of this coupling process, is going to shut down glycolysis in the Krebs cycle and electron transport. Those processes will all grind to a halt because there's nothing to phosphorylate anymore. And because of that, this stops. This gradient becomes huge, and the large gradient shuts down NADH oxidation. NADH builds up. The Krebs cycle enzymes, glycolytic enzymes that generate this, they're going to shut down too. So it backs up the whole process. Experimental evidence for that can be seen with isolated mitochondria. So you can carefully chop up cells in a way that doesn't destroy the mitochondria. And through centrifugation and other techniques, you can get purified mitochondria. So you just grind up a half pound of tissue, you get yourself a nice pellet that's intact mitochondria. And these mitochondria, you can feed them substrates, and you can watch them respire. So you give them something like pyruvate. They'll pump the pyruvate in, they will do the Krebs cycle stuff, they'll do electron transport, you can see oxygen being consumed. So typically what you do is you do it in a sealed chamber.
How long would they live?
Oh, they're good for a day or two. So you have some kind of sealed chamber, temperature controls, you put a stir here on the bottom, and you've got mitochondria floating around inside. And then at the top you put in an oxygen sensor, so you can monitor the oxygen levels inside. And then you've got ports where you can inject in stuff like substrates like pyruvate or ADP. So you can test the effect of limiting ADP or excess ADP and stuff like that on the rate of respiration, because you can monitor how fast the oxygen is being used up. So there's lots of detail to that kind of experiment, but I want to focus in on this area of the box. Of the various conditions, the various experimental conditions, two of them are pertinent to our discussion. Mitochondria can be considered in 5 states. State 3 is the one where it's got everything it needs. It's got oxygen, it's got substrate, that could be pyruvate or malate or any of the TCA cycle intermediates. So let's say pyruvate, that's easy to understand. That's their normal food. So they've got oxygen, they've got substrate to oxidize, and you've given them ADP to work on. They're working at their fastest rate there. They've got everything they need. Stage 3 rates of respiration are high. So under those conditions, the oxygen sensor here gives you immediate readout in real time of what the oxygen level is in there. So if you plot the respiratory rate, the rate of oxygen consumption as a function of time, you would see the rate dropping. The other state of interest is the so-called stage 4 rate, where the only thing that's missing from the mix is ADP. And in that state, the rate of respiration is close to 0, or slow at least, depending on how healthy the mitochondria are. So this would be stage 3, and that would be stage 4. This would happen, this transition would happen if you gave some small amount of ADP, enough to last it for maybe 30 to 40 seconds worth of respiration. So let's say at this point you inject some ADP, you see the rate immediately goes up. Why does it go up? Because now it can do oxidative phosphorylation. Now there's some work to be done by that little engine. So it's in this period of time, it's making ATP out of this added ADP. That activates the electron transport chain. That activates the Krebs cycle. All that pyruvate that's in there is now being maximally consumed. At some period of time, though, it will do its work. It will have converted all that limited, well, let's specify here, it's limited amounts of ADP. When that's all converted to ATP, it goes back to kind of a resting basal rate. So that's showing the coupling of electron transport to oxidative phosphorylation. So here's some preliminary stuff. Here's the state 3 rate in real time. This is kind of data you'd actually see where this is the oxygen level as a function of time. So it's consuming oxygen, it's breathing, if you will, it's respiring at a high rate. The slope of this line is the respiration rate. So here's another state 3 rate, a very steep rate. When you consume all the ADP, it's all converted to ATP. It goes into the state 4 rate. The ratio of these two is something called the respiratory control ratio. It's a measure of the coupling between those two processes. It's telling you how sensitive the respiratory rate is to the process of ATP synthesis. And this ratio, if you haven't beat up the mitochondria too much during your purification strategy, you're going to see ratios of about 10 for good healthy mitochondria.